This video will be a summary of some examples of molecular transport, and primarily we're just going to look at some examples of diffusion and osmosis and what the consequences of these molecules moving back and forth across the plasma membrane can have in terms of volume and pressure inside of the cell. We're going to discuss some of the vocabulary that we covered earlier in the PowerPoint notes. So the processes in terms of molecular transport that we're going to look at, as I mentioned, are diffusion and osmosis. And in general, remember that diffusion is just the movement of molecules from an area of higher concentration to lower concentration seeking equilibrium. And osmosis is a specific type of diffusion that involves the movement of water from an area of higher concentration to lower concentration seeking equilibrium. When we look at these graphics, and basically we just have some examples of concentration gradients where we have a cell suspended in a beaker. And we had talked earlier about what a solution is. And when we look at the movement of these molecules back and forth across the plasma membrane, this movement is occurring between two different solutions. And a solution is made up of two things. A solution is made up of a solute, which is generally a solid or a gas. In this case, we're going to be looking at oxygen and carbon dioxide, two molecules that readily diffuse across the phospholipid bilayer of the plasma membrane. Remember, oxygen and carbon dioxide are both nonpolar. That is, that they don't have a charge associated with them. And so they're able to cross that membrane just based on the concentration gradient. So one part of the solution is the solute, solid or a gas. The other part of the solution is the solvent. And for our purposes, we'll always just consider that solvent to be water. So those two components make up the solution. And one of the things to keep in mind is that the percentage of the solute plus the percentage of the solvent always has to equal 100%. When we look at this example here, basically we have two solutions. We have the solution outside the cell, which we've described as the extracellular fluid. And that extracellular fluid really is basically just anything that's outside of the cell, the solution that's outside of the cell. So that could be blood, water, lymph, whatever it might be. And then we also have the solution inside the cell, which is the cytoplasm. And that's a term that we've heard before. So we have two solutions, the extracellular fluid and the cytoplasm, and those two solutions are separated by this semi-permeable membrane, which we know as the plasma membrane. And we looked pretty closely at the plasma membrane, and we know that that's that phospholipid bilayer with those globular proteins that are kind of randomly arranged throughout that phospholipid bilayer. This first example, really pretty straightforward. The solute in this example is oxygen. And we're looking at basically 18% oxygen out here in the extracellular fluid, 10% oxygen inside the cell. And so remember that during diffusion, that oxygen is going to move from a higher concentration to a lower concentration seeking equilibrium. This is what happens in your cells. And so if you take in oxygen, basically transported by the red blood cells, you get a higher concentration of oxygen outside of the cell than inside of the cell, and that oxygen moves from that higher concentration to lower concentration seeking equilibrium. That's part of what provides basically the oxygen for cell respiration. So that process is just called diffusion. Pretty straightforward stuff. One of the things to remember is that this O2, this oxygen, is the solute. And we'll come back to this and look at the other component of the solution here in just a second. So the O2 is the solute in both of these solutions, in the extracellular fluid and also in the cytoplasm. The second example is pretty much the same thing. And so you have the extracellular fluid outside of the cell, the cytoplasm inside of the cell. As that oxygen moves into the cell and is used by the mitochondria basically to do cell respiration, it generates a byproduct. And that byproduct is carbon dioxide. And so in this example, the solute in both the extracellular fluid and in the cytoplasm is CO2 or carbon dioxide. Here we have 12% versus 6%. CO2 is small, nonpolar, so it will freely move across the membrane. It's going to go from a higher concentration to a lower concentration seeking equilibrium. Again, just basic diffusion. So that's kind of got us started here. But we have this. Remember that the solution is composed of two different things. It's made up of the solute and the solvent. So far we've just kind of been looking at the concentration gradient associated with the solute. But there's always two concentration gradients involved in these solutions. There's a concentration gradient between the solute, the solid or the gas, and the concentration gradient between the solvent, for which our cases is going to be water. So let's look at an example where we take into consideration the concentrations of both of these components of the solution. Here we have a phosphate molecule. And one of the things to know about phosphate is that it has a charge associated with it. So if you look at this, this has this big old -3 charge. And there are a couple of things that will prevent molecules from moving. One is if they're polar or have a charge. Another one is if they're extremely large, things like big proteins or starches or polysaccharides that are just too large to cross the membrane. This phosphate molecule is our solute, both in the extracellular fluid and in the cytoplasm. But now let's consider the other piece of this in terms of the solution. You have a solution outside of the cell, which is the extracellular fluid. And remember that the solute plus the solvent has to equal 100%. So we need to determine the concentration of the solvent. And to do that, you simply take 100 minus 2% and you end up with the concentration of the solvent, which is the water. So in this scenario, 100 minus 2% is 98%, and that's going to be the concentration of the water in the solution in the extracellular fluid. You can do this if you want to, you may not need to, but sometimes when I see examples of this or I'm setting up examples of this on a quiz or a test, one of the things that you might do initially is just, cover up the solution outside the cell and determine the concentration of the solvent inside, and then vice versa. So here we have our two concentrations, the concentration of the solute and the concentration of the solvent in the extracellular fluid. So now we need the concentration of the solvent inside the cell in the cytoplasm. So here we have 4% phosphate. So we take 100% minus 4%. That means that we're going to have 96% water. And the water is our solvent in the solution. This is a higher concentration inside the cell of the phosphate molecule. And so the tendency would be for this molecule to go from a higher concentration to a lower concentration seeking equilibrium. The issue that phosphate has is that it has this charge associated with it. And so it will actually interact with the surface of the membrane. It's not able to freely diffuse. And so because the solute molecule can't diffuse, the solvent molecule is going to move to try to balance this concentration gradient. So here we have a 98% concentration of water outside the cell in the extracellular fluid, 96% water inside the cell. So the water is going to follow the concentration gradient from a higher concentration to a lower concentration seeking equilibrium. 98% is higher than 96%, so the water is going to move from a higher concentration in the extracellular fluid to the lower concentration of water in the cytoplasm. And when that water moves from a higher concentration to a lower concentration seeking equilibrium, that movement of water is a specific type of diffusion called osmosis. So the movement of water is osmosis. And it will move. It'll cross that plasma membrane. Primarily, it will cross the plasma membrane through that channel globular protein that we talked about before, the aquaporin. So let's bring in a little bit of this vocabulary that we talked about earlier. And when we describe basically the concentration of the solutions, we use these prefixes tied to the suffix tonic. And so we use those terms iso, hypo, and hyper to describe the concentration of the solute molecule in the solution. Now when we apply these prefixes, we're always going to apply those prefixes to the concentration of the solute in the extracellular fluid. And so let's add that just out here in the margin. Those prefixes, iso, hypo, and hyper, are going to be used to describe the concentration of the solute in the solution, and specifically we're going to be looking at that, using that description of that concentration for the solute in the extracellular fluid. So let's drop down here and see kind of how we can apply those prefixes in these examples down here. Here this, the solute in this example is glucose, a simple sugar. And so just to kind of keep us on track here, let's go ahead and label our solute, which is the glucose, both in the extracellular fluid and in the cytoplasm. When you get these types of examples, the first thing that I do is to determine the concentration of the solvent. So here if we have 8% glucose, that's the solute. We know that everything else is the solvent, the water, and so we take 100% minus 8%, and we end up with what's left, which is 92%, and that's the solvent. And for us, that solvent is always H2O, water. So I have the concentration of the solute and the solvent in the extracellular fluid. Now I need the concentration of the solvent in the cytoplasm. So here we have 6% glucose. So 100% minus 6% is going to leave us with 94% of solvent in the cytoplasm, in the solution inside the cell. And that is, again, always water for us. Here we're going to see actually the movement of both the solute and the solvent. And so glucose, slightly polar, but it will move, and when it moves, again, it's going to move through one of those transport proteins, that carrier globular protein 1, through that process of facilitated diffusion. 8% is higher than 6%, so glucose is going to move. It's going to move from a higher concentration to a lower concentration seeking equilibrium. That movement of that glucose molecule is diffusion. So that takes care of the concentration gradient on the solute side of the solution, but we also have this concentration gradient going on up here with the solvent. The solvent will move as well, that solvent being water. It also moves through one of those transport proteins, the channel globular protein, but specifically the aquaporin, 94% is higher than 92%. So that water is going to move from an area of higher concentration to an area of lower concentration seeking equilibrium. The movement of water is osmosis. So we're almost home. Now we just have to apply these prefixes of iso, hypo, or hyper to describe the concentration of the solute in the extracellular fluid. Okay, now remember that iso basically means the same. And so that means that the concentration of the solute and the solvent is in balance. It's the same on both sides of the membrane, in the extracellular fluid and also in the cytoplasm. Hypo means lower, like hypothermic. Okay. And then the last prefix that we looked at is hyper. Hyper means higher. Okay, so when we apply these prefixes, remember that we're going to apply those prefixes to describe the concentration of the solute in the extracellular fluid compared to the concentration of the solute in the cytoplasm. So this is the number that we're interested in, right here, this 8%, which is the concentration of the solute in the extracellular fluid. Now when we compare it, we're going to compare it against the concentration of the solute in the cytoplasm. 8% is higher than 6%. And so if it helps you, just draw a little arrow there. And you can come up and look, okay, well, which one of those prefixes means higher? Well, it's hyper. And when we use those prefixes to describe the concentration of a solution, we're going to tie it to the suffix tonic. So this solution out here that has 8% glucose we would describe it as hypertonic. And because both of these molecules can freely move, eventually these two solutions are going to come into equilibrium. When they come into equilibrium or into balance, both solutions will then be isotonic. So this solution is hypertonic, but because both molecules, the solute and the solvent, can move, eventually this solution will be isotonic. Okay, so the prefix describes the concentration of the solute in the extracellular fluid. You tie that onto tonic. Tonic means concentration. And so in this scenario, eventually both solutions will be isotonic. That's not always the case. If we look over here at this next example, kind of start out the same way. Okay, so basically, know that the starch, a solid or a gas, is a solute, both in the extracellular fluid and in the cytoplasm. And we have to, once we get the concentration of the solute, the next thing is we have to get the concentration of the solvent. So out here, we're going to take 100% minus 3%. That gives us the concentration of the solvent in the extracellular fluid. So 100% minus 3% is going to be 97%. And that solvent is always going to be water. Have to do the same thing for the solvent inside of the cell in the cytoplasm. 100% minus 7% is going to be 93%. That's the solvent concentration in the cytoplasm. Solvent's always water. Based on this concentration gradient, you have 7% starch inside, 3% outside. The movement would tend to want to go in this direction through the process of diffusion. The problem is that starch doesn't necessarily have a charge or a large charge associated with it, but starch is a big molecule. It's a polysaccharide. It can't diffuse. So we run into that same kind of scenario that we had with the phosphate molecule in terms of the solute not being able to move. Because the solute can't move, that means that the solvent is going to move to try to balance that concentration gradient. Here we have 7% water outside the cell, 93% water inside the cell. The water is going to move from a higher concentration to a lower concentration seeking equilibrium. That movement of water is osmosis. Again, it's going to occur across that channel globular protein, the aquaporin. And this type of scenario will eventually create a problem for the cell in terms of volume and pressure, because we're never going to get all of this water and this solution into this cell. As that water kind of continues to move into the cell, it's going to hydrate the cell. It's also going to cause the cell to increase in volume and eventually increase in pressure. So let's go back to our prefixes up here. And remember that this is the number that we're interested in. This basically number that is the concentration of the solute in the extracellular fluid. And we're going to compare that number to the concentration of the solute in the cytoplasm. Here we have 3% versus 7%. Okay, and so that number is lower than that number. The prefix that means lower is hypo. So when we describe this solution, we're going to describe this solution as in terms of the extracellular fluid as hypotonic. If you suspend a cell in a hypotonic solution, it basically causes the cell to become hydrated. So this causes hydration of the cell. Hydration basically means it's gaining water. If we increase the volume and pressure enough inside of the cell, eventually this cell could rupture or burst. And if that cell basically ruptures or bursts, that is called lysis. So let's take a look at the last example here. In the last example, we basically have just reversed the concentrations of the solute. So here we have 7% in the extracellular fluid in terms of our concentration of the solute. We take 100 minus 7%. That leaves us with 93% left over for the solvent, which we know is the water. And then we have inside the cell, we have 3% starch, and so 100 minus 3 is going to give us 97% for the solvent. Again, the H2O. The starch would like to go from a higher concentration to a lower concentration, seeking equilibrium, wants to move through the process of diffusion, but it's too big, it can't cross either the phospholipid bilayer or any of the globular proteins. So we're kind of stuck with this concentration gradient between the solvent. The solvent is the water, and the water can move. It's going to move through those aquaporins, and 97% is higher than 93%, so the water is going to leave the cell. It's going to move from the cytoplasm to the extracellular fluid, still called osmosis. Okay. And the only thing that we have left to do here is to describe what's happening in terms of volume and pressure inside the cell, and also describe the concentration of the solution in the extracellular fluid. So we're going to look at this number, 7%, and compare that number to the solute in the cytoplasm. 7% is higher, so we would say that this solution, the extracellular fluid, is hyper. Remember that hyper is the prefix that means higher. And so this would be a hypertonic solution in the extracellular fluid. Okay. Now here you can see that the water is leaving the cell. So this causes dehydration of the cell. And this is going to have an impact basically on volume and pressure inside the cell. As water leaves the cell, volume and pressure are going to decrease. And so, for example, you can see this in plant cells. When plant cells don't have enough water inside of the cell, they lose volume and pressure, and they kind of shrink and shrivel. And the term to describe that condition is the cell loses volume and pressure is called crenation. So you take a look at this entire graphic. Basically, just a few things to remember. We're dealing with two different solutions, a solution outside the cell, which is the extracellular fluid, the solution inside the cell, which is the cytoplasm. It's separated by that semi-permeable membrane, the plasma membrane, and the major function of the plasma membrane is to control what comes into and out of the cell. Some molecules can freely move if they are small and or nonpolar. Some molecules can't if they're polar, or which means basically that they have a charge associated with them, or if they're particularly large. The other thing to take away from this is how do we describe the concentration of those solutions, specifically the concentration of the extracellular fluid, using those prefixes iso, hypo, and hyper, and then be able to relate that to what's going on with respect to volume and pressure inside the cell. And, is that causing hydration of the cell, which in that situation, basically, you would have increase in volume and pressure and could result in lysis. That's what occurs when you suspend a cell in a hypotonic solution. Or is the concentration gradient set up so that you're actually water is leaving the cell, which causes dehydration of the cell, and you lose volume and pressure, and in that situation that would cause crenation. So that's it for our summary of molecular transport and diffusion and osmosis.